Guys, today we are going to discuss regarding the markers and its types. Okay, let's start. First, we'll get to know what is a marker. A marker is any kind of sign. Any kind of sign is said to be a marker. We can define this marker as any kind of mark that should follow two essential conditions. What are those two essential conditions? We will get to know one by one. First one is it should be stable across all the generations. And the second one is it should follow a specific trait. Right? Now we will get to know about, we will discuss about types of markers. Okay, first comes the morphological marker. These markers are again classified into different types. First one is the morphological marker, second one is biochemical marker, third one is cytological marker, and the fourth one is molecular marker. Okay, again let's discuss all these one by one. First one, again this molecular marker is again divided into two types. First one is hybridization based marker, and the second one is PCR based marker. Example of hybridization based molecular markers are rflp. Here we will get rapd, ssrs, scar, aflp, like this, so many. Okay, the another thing is, now we will discuss one by one. First, here comes the morphological marker. The morphological marker is anything that we will visually observe. If we visually observe anything, then that is said to be the morphological marker. Like if we see the difference in between traits based on their phenotype morphologically, then it's said to be a morphological marker, right? This morphological marker is also known as genetic marker, right? For example, if we have any two plants, for example rice and wheat, if we want, if anyone gives us one rice plant and one wheat plant, and if they want to say, if they asked us to say any differences between these two plants, we can easily say, right? Like the same way, this morphological marker, here we are using morphologically, we are saying that that's why it's said to be a morphological marker. For example, if they given any same plant, for example rice plant of two varieties, variety A, variety B, okay, then if they say you the difference, then also you can say the difference very easily morphologically, right? For example, variety A is having the height, for example, take the height, for example variety A is of 30 centimeters, variety B is of 40 centimeters. Number of spikes in variety A is 6, in variety B is 10. Number of panicles, like this, for example 4, for example 3, like this, anything. We can see the differences, we can say the differences between these two varieties morphologically, right? Then like this is used as the morphological marker. For example, size, shape, color, any morphological characters that we can take as a marker. This morphological marker where we will use means this will generally used in this varietal identification. In varietal identification we will generally use this morphological marker. Now comes to the advantages and disadvantages of morphological marker. Okay, what we can see, we can easily observable. We can directly measure the phenotype. We are directly measuring, right? And no specific technology is used because we are generally seeing with this naked eye, right? No specific technology is used. For example, while coming to the disadvantages, dominance. For example, if any genotype is having, for example, okay, this plant will be tall as it's having both capital T, capital T. This is having small t, dwarf, and this is also having capital T, so it will be tall. We can't differentiate between this first and third plant, right? One is homozygous dominant and one is heterozygous. That's why we can't difference, like dominance plays a key role here. If any dominance is there, that character will only express if that is dominant. That's why, like, that is one of the disadvantages present here. Okay, and the second thing is pleiotropy. If epistatis is present, that also we can't observe. Less polymorphism. What polymorphism actually means? Ability to have different forms is said to be the polymorphism. We will discuss it with an example later. And then another thing is highly influenced by environment, and it's few in number. Due to these many disadvantages, the scientists have went into the biochemical marker, which is our second marker. Means first one is completed. We have discussed the advantages and disadvantages, what actually the morphological marker means, and now we are going to the second one, biochemical marker. This biochemical marker is also known as protein marker. Like if there is any change in DNA, it leads to the change in protein. That's the trait. If any DNA is there, DNA gives rise to protein, protein gives rise to character, right? If anything in the protein, if any DNA, if there is any change in DNA, that leads to the change in protein, that leads to the change in character, right? By studying this protein profile, means study of this genome, we are identifying this biochemical marker. See, it's related to variation in protein and amino acid binding pattern. And the disadvantages here is cytological marker. First one and second one is completed. Now the third one is cytological marker. What actually cytological? Cyto means cell, right? Based on as the name depicts that, based on cellular features. For example, staining technique. For example, Geismar stain, staining, or any cytological techniques if we use. For example, fish gish, fluorescent in situ hybridization or genome in situ hybridization. If we use any techniques, cytological techniques such as this, then also it's said to be the cytological marker based on the cellular feature we are observing this, right? And here comes the main one, molecular marker. Okay, now the molecular marker is the most advanced kind of markers, like, and again it's, as we have discussed, it is divided into two types. First is hybridization and PCR based markers. We'll discuss one by one. Where this molecular marker is generally used, for example, if we are having any variety, for example variety A, okay, and two plants we have taken, plant 1 and plant 2. If anyone wants to if anyone asked you to say the differences between these two plants, then you can't say the differences because they both belongs to same variety, and normally the seeds we will get, which we will get from the same variety, are homozygous in nature, like homogeneous population will be there, so the entire population is same, so you can't even say what are the differences among them, right? In this case, molecular markers are useful. Say here differences. This difference generally means variation. Identifying this variation is difficult, right? Like if any variety is having variety A and we are having two plants, and variation, identification of this variation between these two plants is very difficult, right? Here comes this marker, for example variety A and variety A, both are same varieties. For example, this is plant, yeah, this is plant A, plant B, like 1 2. We will extract the DNA from this plant, and again we will extract DNA from this plant also, right? If any changes in their DNA structure or any nucleotide sequence, then this is said to be the polymorphism. Like what we will say, any variation in the DNA sequence that is present, this is said to be the polymorphism. Here I have said like here, like I will explain it later. Here I have drawn the diagram, and if any difference between this, if any variation in between this DNA sequence, then it is said to be the polymorphism, right? And what actually molecular marker book definition is: unique DNA sequence used in DNA hybridization, PCR and restriction mapping technique to identify that sequence is called DNA marker. For example, if any DNA sequence is there, okay, in case any, like say A, B, any two sequences, here we will keep our thing as marker, means any particular loci we will take this as marker. That should be, majority of the cases that should be stable, like it should be the same, then only we will take it as marker. We can say this as present in the genome at specific location, which is easily identifiable, is said to be the marker, right? Again, this marker, like we'll discuss what are the characteristics of an ideal marker. Okay, and based on inheritance pattern, again we will discuss it. First we will discuss based on inheritance pattern. Okay, based on inheritance pattern, it's of two types: dominant marker and codominant marker. If any one form of trait that is associated with the marker, then it is said to be the dominant marker. If both the forms of the traits that are associated with the marker, then it is said to be the codominant marker, right? It is like whichever character is dominant, it will show that thing only, right? Here we can easily differentiate between homozygous and heterozygous. In this previous videos, I have clearly explained what homozygous and heterozygous, what homogeneous and heterogeneous, like these four terms very clearly. You can just like take a look from there if you have any doubt, right? What actually dominant marker, how it will be, right? I think all of you know parent 1 and parent 2. If we cross, we will get this F1 plant, right? This is the thing. If we extract the DNA from parent 1, parent 2 and F1 progeny, right? Parent 1, parent 2 and F1. If parent 1 DNA is observable here, parent 2 is here and F1 is here. For example, for any plant character, for example height, right? If parent 1 is having capital T and parent 2 is having small t and the F1 will have capital T small t, right? This is tall, this is dwarf, and this one will also be tall, right? Here you can observe the gel here. Parent 1 is here, parent F1 is also here for this character tall, right? These both are at the same place and the P2 is of at the down. This is dwarf, right? This is small t, capital T, this is capital T small t. Here it is heterozygous, but here we can see whatever the parent 1 is expressing, same thing the F1 is also expressing. Only the dominant character is expressing here, so it is said to be the dominant marker, right? Now we will understand regarding this codominant marker. In this same codominant marker, same P1, P2 and F1 we will take. Parent has P1 has banding here, P2 has banding here. The F1 will have banding at both the places, right? This is codominant marker. Examples of codominant markers are rflp, str, ssr. Okay, here comes the examples of dominant markers are issr, aflp, rapd. These are the examples of dominant marker. Here comes the question, which marker is suitable for us, right? If dominant character is only shown, then in morphological marker also we can directly see for some characters or traits what is dominant, what is recessive, right? That is the disadvantage there. So codominant marker is very useful for us. So we can consider this as best marker is our codominant marker. Here are the examples of the codominant marker. Here comes what are the characteristics of an ideal marker. Okay, the first point is any marker should express high degree of polymorphism. That is, marker should means each marker loci should have two or more alleles at that occur at like some frequencies in that population, right? Second point is marker loci should be extensively distributed throughout the genome. Means marker loci, they should be present, like distributed all over the genome, right? Third thing is codominant in nature, because it provides more information compared to the dominant markers, so it should be codominant in nature. And the fourth one is highly reproducible. For example, if you have taken DNA of any one plant, right? If you test this in, like if you have applied markers in this, the result should be same throughout any laboratory or different experiments, like whatever the laboratory you have taken or any place you have taken, it should be same result should be coming for this both the laboratories or the thing. Like it will like if different information comes, it's not useful, right? Like everyone will have different results, so the marker which you are taking, that should be highly reproducible, so that the results won't differ from different laboratories or different experiments in the same laboratory, which can be easily comparable, right? And fourth point is 1 2 3 4 fourth point complete. Fifth point is detection of markers. It should be easy, rapid and simple. Sixth point is the cost of detection. It should be minimum. Seventh point is the detection system should be automated for high throughput genotyping. Here we can say amenable to automation. High throughput genotyping means so many samples can be processed at the same time. Means many samples we can do it at the same time. Then this is like this leads to the more increase in results at the with very less time, right? And another point is pleiotropy. We can't say pleiotropy should be absent, like it's not possible, so it should be minimum. Epistatis should be absent. Environment effect should be absent. These are the 10 points for the characteristics of an ideal marker. Next class we will discuss regarding each marker clearly, what are its applications and all, or we can discuss the applications in this only, and then from next class we can go to the like molecular marker systems and different types of markers. Now we'll discuss here applications where normally these markers have been like generally used, right? Genetic diversity. When we are assessing the genetic diversity, these molecular markers have been used. Second thing is genetic linkage map. In linkage map construction also we will use this. Third point is identification of new alleles in the germplasm. If any germplasm is there, if any new alleles are present in that, we can identify that using that molecular markers. We can say identification of new alleles. Fourth point is marker assisted selection. We are doing selection based on the mark with the help of markers. This is said to be the marker assisted selection. We can have one detailed lecture on this marker assisted selection. And again, where these molecular markers have been useful means it's useful in crop evaluation. How it is evolved and everything, we can study that using these molecular markers, right? Thank you.